And then we have the practical this afternoon. So I can figure out how to run this. So an overview of what I'm going to do. I have two presentations. The first one, which will be before the break, is going to be talking about genomic sequencing, a little bit about the past, what's going on at the moment, and looking forward into the future. After the break, I'll talk mostly about RNA-seq and how this informs us about Leishmania biology. And then after lunch, we're going to have a practical session where you guys will learn how to use TriTrypDB, do some pathway analysis, and use a site called TDR targets. So that'll take about 3.5 hours this afternoon. And I'm going to sort of let you work your ways out. And we'll talk about that when we come back after lunch. OK, so I'm just going to give you a little background on genome scale analyses, or Omics, as they're called. Really, there's sort of four different technologies that we can use. Genome sequencing, which really tells you about the gene content, but also can tell you about modification of the DNA as well. So that's epigenetics. Mass spectrometry can tell you about protein abundance and phosphorylation. You can also actually look at metabolomics using mass spectrometry, which I should put on this slide, I guess. Microarrays, or RNA-seq now, which is really mostly replaced microarrays, will tell you about messenger RNA abundance and alternative splicing. And then there's a relatively new technique called ribosome profiling, which is useful, first of all, for re-annotation of the genome back here because it improves the gene predictions, but also gets at translational regulation. So using these techniques, you can basically use this to tell you what genes are present. You can then use the RNA approaches to figure out what their RNA levels and how those are regulated. You can use ribosome profiling to work out how those are translated. And as we'll see this after the break, there's not a particularly good relationship between RNA levels and protein levels, which you can measure here. You can also measure, with mass spectrometry, you can measure post-translational modification. So then, if you add on to that the metabolome, you can figure out what these proteins are actually doing. So you can have a really good view of the entire organism and really pretty much everything that's going on in the organism, although it's not quite that easy in practice. OK, so sequencing. This is what we did when I started the genome sequencing projects for Leishmania and trypanosomes in the mid to late 90s. We used a map first and then sequence approach. So you basically cut your genome up into a bunch of big cosmids or back clones, and you then used restriction mapping or hybridization to fingerprint them so that you could come up with a map of how all of these clones overlapped, and then you would choose a minimal tile path out of these, and then you would shotgun sequence the clones, because back in the 90s, it was a big task just to sequence a single cosmid, which is about 30 kilobases, or a back of 100 kilobases. So that was really all you could do there. So it was slow, but one of the advantages was that it was accurate because you were dealing with small DNA. And it was also scalable. So you could actually take these, after you'd done the mapping step, you could take each of these cosmids and you could distribute them around, and everyone would start sequencing a cosmid. And in fact, my first visit at Trieste was because in the Leishmania project, there was a ULeish consortium that involved a lab here at ICGEB. And so they sequenced half a dozen cosmids, and other people in the consortium sequenced half a dozen cosmids, and they could sequence them and annotate them. And you could do that because you were doing them one at a time. But it was really slow and expensive to do that. So that soon gave way to a map-as-you-go approach, where rather than doing the restriction mapping, you basically just took your clones and you end-sequenced them, and then you shotgun sequenced a few of them, and then you used the end sequences to figure out which ones had the least overlap, and you went on to the next one. So it was a little bit more rapid because you didn't have this sort of startup phase, but it was still scalable and divisible. So that was sort of in vogue in the late 90s, probably. By the the early 2000s, we started doing chromosome shotgunning and then whole genome shotgun, where you basically made small and large insert clones and you sequence them and you threw all of the sequences into a computer and you asked the computer to do the assembly, and it did. And the reason why this took so long to take off was because it was only by the early 2000s that the computational power was sufficient to be able to do this. The problem with this from a genome or from a consortium perspective was it really wasn't scalable. You basically got no results until you'd done all of the sequencing. And so really this put all of the sequence being done in large genome centers, which is really where it's sort of lived ever since, until recently now that everyone can have their own little sequencer, which we'll talk about in a moment. So once you've got your genome assembled, you have to actually do gene prediction and annotation. So the gene prediction step is relatively complicated, depending on your organism. If you're working in Leishmania, it's actually not all that difficult. First step is to find all of the open reading frames, but you have to remember that not all open reading frames make proteins, and so they're not genes, but that's the first step. If it's not an open reading frame in Leishmania, it's not going to be making a protein. You then use statistical methods to determine the likelihood that each of ORF makes a protein or not. And so I like to use about 4 different approaches. You can use a codon bias. You can use a nucleotide bias with a program called GeneScan. You can use the period 3 constraint using a very old piece of software called testcode. And then we used to use Glimmer, which is a hidden Markov model. And there are a couple of other newer techniques that have been available, mostly in the Markov modeling. So I like to use all 4 or 5 of these and then do a consensus approach. If 3 out of the 5 say that this is a gene, then we'll call it a gene. And if only 1 out of 5 says it's a gene, we won't call it. And so we built a little algorithm or a little program that enables us to do this. It's called Magi. I had a student do that many years ago. Once you've identified what you think is a protein-coding gene, you then need to search the sequence databases to find something that looks like it, and then you can give it an annotation. If the protein looks like another protein that has a known function, then it's likely that yours has the same function. And so BLAST is really the sort of tool of choice for this, although there are other methods that involve clustered ortholog groups. And you'll be doing a little bit of that this afternoon if you haven't already done it. Probably most everyone here has done a BLAST search or 2 anyway. So that works for about 40% of the genes in Leishmania. They have some similarity to something else, but the rest don't, at least using BLAST. So we can use various different motif finding programs like patterns or profiles or domains that will identify short regions that gives you some hint about what that protein might be doing, but it probably won't really tell you its function. And then finally, back in about 2000, this consortium called the Gene Ontology Consortium was put together to really use a systematic and defined language to describe the function of genes. Prior to that, it was sort of the Wild West where whatever the annotator felt like on that particular day, that was the description that they used. And so it became really very difficult to compare the same gene in more than one genome. And since the advent of gene ontology, or GO, it's really much easier now to know that you're talking about the same thing in 2 different organisms. And then you shouldn't forget that there's also RNA genes that you can identify using tRNA scan for tRNAs or BLAST searches for other types of RNAs. A lot of the smaller RNA genes are actually very difficult to predict computationally, so you actually have to use experimental evidence to identify those. Oh, and a bottom part here that's getting cut off is that you can do all of this in an automated fashion. Now you can write an algorithm that will go through and do all of these sequentially and make its own calls. or you can do it manually. And obviously, if you do it in an automated way, it takes a lot less time, but it's not as accurate as doing it manually. So basically, what we generally do is make a whole pile of automated calls and then slowly over time curate those so that they become more accurate. So the TriTryp genomes were published in 2005. There were 3 genomes in this initial lot, hence the name TriTryp. L major, which I had a big role in doing. T. brucei, which I was less involved in, and then T. cruzi, which again we had a fairly large role in doing. So all 3 of them were finished at the same time, though, although I have to say that L major was really the only one that was truly finished. The other ones were more or less finished, probably less in terms of T. cruzi and maybe a little more in T. brucei. And in fact, neither of these 2 genomes is finished at the moment. I'd say Leishmania major is by far the best assembled genome of any of the trypanosomatids, even to this day. Although I will tell you that you'll see at the end of this, probably L. tarentuli will be the best assembly in the near future. And so that enabled us to do metabolic maps of each of these genomes. And in this particular example here, I've sort of zoomed in, and what I'm going to show you in a moment is that there are organism-specific metabolism that you can identify just by looking at the genome. So if you look here, all of these enzymes and pathways are present only in Leishmania. So a lot of Leishmania-specific pathways, but a lot of shared pathways as well. All right, so the conclusion of all of that was that Leishmania had a pretty simple genome, at least compared to the other trypanosomes. There were chromosome pairs that were pretty much similar in size. Sometimes they were slightly different because of sub-telomeric repeats. There were very few heterozygous alleles, like less than 0.1%. There were some, but not very many repeats. There are a lot of gene duplications. There were some protein coding genes that had tandem amino acid repeats. And then there was a series of intergenic interspersed repeats that were identified by Mark Willet's group and Barbara Papadopoulou, who did some biology on them. That became apparent only after, we knew that the repeats were there, but we didn't know what their function was until after. And Mark's group has really shown that these intergenic repeats are used to amplify different regions of the genome on a sort of stochastic basis, and then those can be selected. And so we'll come back to that in a moment, some examples of that. So that was frequent gene amplification that used these interspersed repeats. The organization of the trypanosomatid genomes is unusual. This is the L major genome. There are 36 chromosomes. I'm sure you all know that. If we zoom in and we look at some of the smaller chromosomes, what you'll notice immediately is that the colored rectangles are protein coding genes, and you'll immediately notice that they are not randomly distributed between strands, which is generally the case in most other organisms. In trypanosomatids, there are these clusters of genes all on the same strand, and so that they're transcribed in polycistronic fashion. And they can be sometimes up to 300 genes on the same strand. They can be organized divergently. On chromosome 1, you'll see that there's a transcription would initiate here and go in that direction and in that direction, so that's divergent, whereas on chromosome 4, it's convergent. And on the larger chromosomes, you have both divergent and convergent clusters on the same chromosome. And often, the convergent strand-switch regions have tRNA genes in the middle of them for reasons that will become apparent later, maybe if I've forgotten whether I kept that in my talk or not. Okay, so that was then. This is now. There in the, 3 or 4 years ago, there was an enormous explosion of new sequencing techniques called next-generation sequencing, or now called second-generation sequencing, because we're onto the third generation now. I'm really going to tell you about the Illumina platform, which is really the platform that's sort of taken hold and now accounts for the vast majority of sequencing that's done. So with Illumina sequencing, you do relatively short reads. Now you can do up to 300 base pairs, perhaps even a little longer. It's pretty accurate. The big advantage is you can get 3 billion let's say 3 billion reads per run. So on a single machine, you can get 3 billion reads, right? So that's a lot of reads. Takes you 1 to 10 days, depending on how long your reads are. And they only cost as little as 5 cents a base to do this. Actually, 5 cents for a million bases. So the price drop from $2,400 per million base using the old Sanger sequencing to 5 cents is really amazing. So essentially, this technique now means that you can sequence anything that you wish and get huge amounts of data in no time at all. So for instance, it took us, I think, about 10 years to sequence the Leishmania major genome, and now you can sequence the Leishmania major genome in a week.
Compare the whole, you said it was more accurate the way to do it before. Have you compared L major previous sequencing approach with the Illumina to see how
yes.
accurate it is?
So if you do Illumina sequencing and you do assemblies with Illumina sequencing, you will end up with genomes that are like the L infantum genome, the L donovani genome. They are 95% accurate, but they have assembly errors and gaps in them. So you can't get a closed genome by this. You'll get a draft genome. Okay, so I forgot that I was going to say. You get a draft genome. This is also very useful for doing things like RNA-seq, where you really don't need to. Your point is not to try to get an entire closed genome, or ChIP-seq, chromatin immunoprecipitation sheep. So basically now it's cheaper to sequence stuff than it is to do investigate it in any other way. The actual price of this on an Illumina machine is about $2,400 in a lane. You usually, depending on which machine you have, you can get 8 lanes per machine. And you can run anything up to 8 or 10 or 16 samples in each lane. So if you multiplex it, you can run a sample of genomic DNA or entire RNA for $600, and half of that is the cost of making the library. So for $600, you can sequence an entire genome of Leishmania. Then you have to have someone who will assemble it and annotate it, which is going to cost you more than that, probably. Same with the RNA-seq that I'm going to be talking about after the break. Okay, so now it's relatively easy to do resequencing. So you just shear the DNA, and then you put on some primers, and you amplify it up if you need to. You don't even need to do that. And then you put it in your machine, and you sequence it. And you get a couple of 100 million reads for each lane. So that would give you 600x coverage of Leishmania. If you multiplexed it, you can get 50 to 100x coverage, which is plenty to get complete coverage of your genome, other than a few difficult-to-sequence regions. You can use a couple of different programs. We like to use Bowtie 2 to align in each of these short reads back to the reference genome. So as I said, generally this is for resequencing where you already have a reference genome, and what you want to do is you want to compare your genome to that reference. So you don't need to assemble it. You've hopefully got a good assembly to start with, and you just map each of these reads onto your reference, and you can measure things like the changes in Ploidy or somy. You can look at copy number variation, and you can look at single nucleotide polymorphisms. And I'll show you some examples of those in the subsequent slides. You can do a reference-guided assembly where, rather than trying to do a de novo assembly, which you could try, but as I said, it does have its problems, you can assemble your reads based on the reference, and then have a new assembly that has the changes in the new genome. So the problem with the assembly of unallied reads is that you'll end up with gaps, but you can use this to fill gaps that you might find if your reference has gaps in it. Obviously, you won't be able to align to those gaps, but if you have unaligned reads, you can maybe close some of those gaps. So what we've done in our lab over the last few years is done several isolates of L major from actually L major Friedland, which was the original sequenced one. We've resequenced that from DNA in my lab and from Steve Beverly's lab, and found that they've diverged somewhat over the years. We've also sequenced a Sudanese strain from Mohammed Mukhtar's lab. We've done some donovani that we're using in a collaboration with Dan Zilberstein, 2 Sri Lankan strains of donovani in a collaboration with Greg, and I'll show you a little bit of that in a moment, and then some Sudanese isolates. Have a project with Carlos Costa where we sequenced 10 Brazilian isolates of L. shugasi, and we sequenced an infantum from Sudan as well. So most of this is unpublished yet. I think out of all of that, there's only one that's actually made it into a press. Now, this is the typical type of result that you will get, and I should say that the 20 genomes that I've put up there is really very small compared to what's happened at, say, the Sanger Center, where in a collaboration with Jean-Claude Dujardin, they've sequenced 200 isolates of L. donovani from Nepal and compared them. And that work is published, but they got very similar results in terms of what I'm going to show you here. So this is, if we look at a couple of L. donovani isolates, up here these are several different isolates, and what we're doing is we're just measuring on a heat map what's the copy number for each of these chromosomes. So essentially, if the chromosome is colored green, that means there's fewer copies than the 2 that you would expect, and if it's colored red, there's more than 2. And so what you'll see to start with is that chromosome 31 always has between 3 and 5 copies. So there's always more copies of chromosome 31 than any other chromosome. Chromosome 31 is also strange for other reasons. It's got a much lower gene copy number, gene density than any of the other chromosomes. So something strange about chromosome 31 that we don't understand. In addition to that, even though it's actually got more copies of the chromosome than most of the other, than any of the other other chromosomes, the RNA levels aren't as high as you would expect based on the number of chromosome copies. So maybe that's why it has 4 copies, because it just doesn't make as much RNA. Chromosome 23 in the isolates of donovani that we looked at always had 2 to 3 copies, and it was usually not exactly 3, it was usually between 2 and 3. That meant that essentially in that particular isolate, some of the cells had 2 copies and the others had 3. Sometimes you can find isolates that only have 2 copies, but often chromosome 23 has a 3rd copy. These chromosomes here have different copy numbers in different strains. So essentially the take-home message here is that you see a lot of aneuploidy depending on which strain that you're looking at. Sometimes that can be related sort of to some extent to a phenotype. So in this L. shugasi study, what we found was that, as usual, chromosome 31 would have its 4 or 5 copies, and in fact, I think out of the 10 strains, 9 of them had 4 copies and 1 of them had 5 copies. A few of the other chromosomes have got 2 or 3 copies, right? And so they seem to sort of be scattered around. And what we find was that there was a modest correlation between having additional copies of chromosomes and disease severity, but it wasn't a one-to-one correlation. So these are organized here so that I think there are 8 samples in for each chromosome, and the ones that are on the left are from more severe disease, and the ones that are on the right are from milder disease. And so you can see that, here you can see often you get more amplifications associated with strains from severe disease, but not always. And you sometimes see amplifications on ones that come from less severe disease. So I think we need to do more isolates to really see whether this holds up. Also, what's known is that this copy number can vary just whilst growing the strain. These were all isolated, these DNAs were all isolated from promastigote cultures that had been cultured from the patient. So we don't know what this looked like in the patient. In fact, Jean-Claude Dujardin's group has got some evidence that the copy number might go up sometimes if you're looking in amastigotes relative to promastigotes. If we go in and we look at the subchromosome level, so this is essentially a plot here of an entire chromosome, and I've forgotten which one it is now, and this is data from the work that we did with Greg. Oh, this is chromosome 23. You'll notice that over in this region here, the copy number, and essentially what we're doing is we're measuring the copy number on this axis here. And for most of the chromosome, the copy number corresponds to 2. You'll notice it drops off at the ends, obviously, because the assembly that we were using didn't have good sequence at the end. So obviously, we couldn't align any reads to it. In this region here, one of the 2 isolates has an increase in copy number of several fold. And this region corresponds to a previously identified region called the H region, which had been implicated in a number of different drug resistance strains. You'll also see that there's another region over here that's amplified in both of the isolates. It's a little bit of a smaller region. So essentially what we're doing here is the way we did this is we looked at the read density in 10 kilobase windows across the entire genome, and you can identify from plots easily these amplified regions. Here's another one from chromosome 16, which is actually the opposite way around. The red isolate had the amplification here. So this was, rather than being episomal amplification, it looked like this was tandem increase in a series of tandem repeats of a hypothetical gene. So I think, Greg, we haven't really looked at those in any detail yet, have we? So it's the problem with genomic data is you have so much data, you never get around to actually looking at it all. We can also actually do that type of analysis in a different way. Rather than using 10 kb windows across the genome, we can look at the read density for individual genes. So this is going to tell you for each gene what the copy number of that gene is, of course, relative to the reference. So if the copy number in the reference is wrong, you're going to get the wrong answer, but at least you'll have a relative answer. So here we found that these genes here were the ones that I showed you before. These were, I think, from chromosome 23, and these are from chromosome 16. And you can see this is just 2 replicates of the same isolate. We sequenced them twice. You can see that these guys have higher copy number in this this isolate here, whereas the guys on 23 had a higher copy number in this one. But you'll also see that there's an individual gene, a single gene here, which encodes an ABC transporter, which has higher copy number just in this isolate. And we didn't really find that as clearly when we looked in the 10 kb windows. And here's another one, a single gene that appears to be over-amplified or has higher copy number, and this one's on chromosome 35. It's got higher copy numbers in this isolate. Okay. The other thing you can look at is even at a lower depth is you can look at single nucleotide polymorphisms, or also indels, insertions and deletions. So you can look for changes that aren't a result of copy number variation, but they're differences in sequence. And there's software that's available to identify these, but then it's usually a good idea to go back and look at them visually. although you can't usually look at them all, because if you look at any particular genome, you're going to find tens of thousands of SNPs in any genome. So obviously you can't look at them all. You'll want to find ones that look like they are interesting. They cause a change in the protein sequence, or et cetera. Some of them will turn out to be just sequencing errors. So here you can see that these little red dots here are basically differences in nucleotides, but that's probably just because that read wasn't a very good read. So those would be ignored by the software, and we wouldn't count them. And that's where having a good read depth is helpful. This region here, you can see that these reads down here have a particular nucleotide which I can't read, but it's different from the reference, whereas these reads up here in exactly the same position have the same sequence as the reference. So this is a heterozygous SNP. Half of the copies have one base and the other half have the other base. Whereas over here, this is a homozygous SNP where both copies in this particular genome are different from the reference. And so your SNP callers will identify these differently. One thing that's a little bit tricky is that your reference is always going to have a single allele, and so we don't know whether both of these heterozygous sequences are present in the reference unless we've also sequenced the reference. So, and the other dirty little secret is that the reference will not be evenly divided into two allelic groups. There are methods of phasing, but essentially it'll be a sort of a random mixture. The other thing that you can see from this is something that we've already talked about, and you can see the read depth here is a lot higher, and that's because this region is repeated. And so you've got a whole pile of reads stacking up in this region. Probably means that the reference assembly wasn't good in this region. Either that, or there's more copies more copies of this particular region in the genome that was sequenced. If you plot those SNPs and you zoom out to a much higher scale, you can see this is just a portion of one particular chromosome. I can't remember what chromosome it is. So this is 20 kilobases at the beginning of one of the chromosomes, compared to the L infantum reference genome. And what we can see is we've got L. Chagassi, several different strains of L. Chagassi, and you can see that they are very similar to the reference, as you would expect, because Chagassi is similar to infantum, although there are some differences between strains. So for instance, this strain here doesn't have a SNP. It matches the reference at that position, probably. And here you can see that there are some differences right in here as well. So you can see some differences in SNPs between these different isolates. You'll then see that the next most similar to this reference is the L. donovani from Asia, which is actually from Sri Lanka. And then the L. donovani from the Sudan was really quite different. So I found this result a little bit strange, because this, to me, would indicate that this L. donovani these donovani from Asia were actually more similar to infantum than they were to this donovani from Sudan. So I need to follow up on that a little bit more. This is the type of information that could be useful, and again, this is from the work that we did with Greg that's been just recently published. If we looked at the copy number between these two Sri Lankan strains, one of the Sri Lankan strains was isolated from a patient that had VL, and the other was isolated from a patient that had CL. And in fact, most of the Sri Lankan L. donovani seems to cause CL, which is, of course, unusual. When we looked for differences between the two, we found some clear differences in the A2 locus, which is a repeated locus on chromosome 22. And what you can see here is that the copy number of this region in the VL strain was higher than it was in the CL strain. One of the reasons why this is a bit jumpy across here is that this region was actually very poorly assembled in the reference, and I had to go in and reassemble it myself, and I probably didn't get it exactly right. Plus, there are repeats in each of these, amino acid repeats in each of these A2 genes. So that can vary between strains as well. So there's a bit of jumpiness there. But I think it's clear that the VL strain has more copies of the A2 gene than the CL strains. And Greg confirmed that when you look at the protein level. You can see in the CL there's really only 3 bands when you look at a Western blot, whereas the VL has at least 5 bands. And what Greg did was then he knocked out the A2 locus in the VL strain, and it became less virulent. So a pretty striking phenotype in mice. And conversely, if you put a single A2 gene from the VL strain back into the CL strain, it becomes more visceralized. So it's clear when you combine this with some data that Greg had previously generated in, I've forgotten what strain of donovani you'd used initially. Greg, was that the Sudanese strain? So in that case, also implicated A2 in visceralization. The other thing about the A2 gene is the A2 gene is really absent in L major, which is sort of the first hint as to that's had a role. So I think it's clear that the A2 gene or genes has some role in visceralization. Now it doesn't account for all of the differences. There were a few other differences that seem to be important as well, and I can let Greg talk about that because he knows more about it than I do. So essentially, the lessons that at least I've learned from doing this so far is that you have to be careful of sample contamination. And the reason why I say that is that some of the libraries that we made didn't match the reference. And when I blasted, I had 2/3 of the reads in the library didn't match Leishmania. And in one particular example, they turned out to match Burkholderia. So obviously the cultures had been contaminated with a bacterium. There were some Indian strains that we isolated, that we sequenced that were supposed to be L. donovani. They didn't match our reference very well either, and they were Leptomonas, yes. So those Indians in the audience, be very careful about things that are marked as L. donovani because many of them are actually now Leptomonas. Whether or not those Leptomonas came from the patient is, I think, not entirely clear, shall I say, at least in all of the isolates. I'd like to talk to you a little bit more about that. I think there is some likelihood. So the reason why I warned about contamination is that it's clear that Leptomonas seems to grow better in culture. So it may be that some cultures are contaminating other cultures, which is sort of reminiscent of the HeLa cell story in humans, where at one stage practically all of the cell cultures, human cell cultures in the world were HeLa cells, even though they weren't supposed to be. Anyway, so you do need to be careful about that. Second lesson was that there are extensive changes in somy between isolates. Whether these have any biological function other than just being adaptations to grow in culture remains to be seen. There's some indications that at least some of them are important, but sometimes they probably won't be. There's lots of smaller copy number variations that, again, may or may not have a biological role. Sometimes, so it's going to be hard to disentangle those that are just sort of randomly there from those that actually do have biological function. So I think you have to be careful in both of these cases about drawing conclusions where you've only got a small number of samples, and if you compare, let's say, a sensitive strain to a resistant strain, and you see copy number variations, be it at the whole chromosome or subchromosome level, saying that that's actually associated with the resistance or sensitivity phenotype. With a small number of strains, you have to be very careful about that because you'll have this sort of random background noise that's not associated with the phenotype that you're looking at, but then there may be some association. And so you have to do enough samples to really be confident that there's a clear association between the two. And then there are lots of SNPs. And I said the problem with SNPs is that usually there's tens or hundreds of thousands of SNPs in any, but when you compare any genomes. And so sorting out of that which ones might be important is a real task, because clearly you can't do experiments on hundreds of thousands or even tens of thousands of different SNPs. So you have to prioritize which ones to look at. So SNPs that cause frame shift mutations in a protein coding gene are probably the first thing that you would look at. And then you would secondly look at those that cause non-synonymous changes, i.e. they change the amino acid sequence. Probably you would ignore SNPs that were in non-coding regions, but that doesn't mean that they're not important. Okay, so that's sort of a sum up of what I've just said here, is that it's difficult to associate any of these changes with differences in pathogenesis or disease outcome. Even when you sequence hundreds of genomes, which was done in the Nepal study, there was not really clear lessons that could be learnt from the genome sequence there. It wasn't to say that there weren't some lessons that were obvious. So you have to be careful, and you have to think about what you're trying to do here before you go out and sequence this. The other lesson that I didn't put up here is that this is not something that you can do in your own lab. You really have to have a group of bioinformatics people standing behind you to do all of the analysis that is required to do this, because it's not trivial. Just handling the data itself is non-trivial. Each Illumina data set probably, when you get it off the machine, is a couple of gigabytes, and then by the time you've done the analysis, you're probably using 10 gigabytes per sample. And so if you have 100 samples, you're up in the terabyte range, and that's expensive. In fact, at one stage, I think Matt Berryman at the Sanger Center said that it was actually cheaper to resequence the genome than it was to keep the sequence results. And that's actually true. You basically sequence the genome and then you throw away all of the data except for the final analysis. And it was actually cheaper to do that rather than it was cheaper to resequence that sample than it was to keep the data. Okay, so what's next? What are we going to look forward to next? Okay, well, the first thing is we're going to see lots of genomes. Then I'm going to talk about a new technique called smart sequencing, or single molecule real-time sequencing. And then finally start talking about single-cell genomics. So rather than sequencing a population of cells, and that's why you get this sort of 2.3 copies of a particular chromosome, let's look at individual cells and look at what their genome looks like. The same will apply this afternoon when I talk about RNA as well. We aren't there yet. We are there for this. And as you can see from this slide, these are all of the different genomes that have been published for each of the 3 different trypanosomatid species. Actually, there's more than this. Is African tryp, so there's more than just brucei in there. Spend a little bit of time talking about PacBio sequencing. Similar to Illumina sequencing, you don't need to clone. The difference is that you generate a smaller number of long reads. And these, by long, you can get up to 20 kilobase reads, a single read, coming off a single molecule. And I don't have time to go into why this works. I'll explain it a little bit. But the fact that you get very long reads means that you can get much better assemblies, because you essentially get reads that are long enough to go through all of these repeats. And so you'll know, the algorithm will know how to assemble those sequences much better than it will if you've got a bunch of small repeats that could go in multiple places in the genome. The other advantage of smart sequencing is that it actually sequences and tells you which bases are modified and what they are, and I'll explain that in a moment. The costs about $500 to do a single chip or a single smart cell, and you need probably about 10 of these smart cells to sequence a Leishmania genome. So it's about $6,000 to sequence a Leishmania genome, which is 10 times the price of the same coverage from Illumina, but you get a much better assembly. So I think that PacBio is going to be really good to get really good, accurate reference genomes for every species of Leishmania, and then we'll use Illumina sequencing to resequence all of the strains and find the differences. So that's where I think we're headed over the next couple of years. So
for the genomes that we have problems, for example, thick genome,
mm-hmm.
it was very difficult to assemble.
Mm-hmm.
Now the stem-fly genomes are
mm-hmm.
difficulties assembled. Then you would recommend this type of...
Smart sequencing should work on those, yes. It's really very good for repetitive genomes. Now, that's not to say that it will solve all problems. There's another problem that we had with T. cruzi, and that is that you have this hybrid genome. So there's a lot of difference between alleles. And worse than that is that some part of the genome, the alleles are similar enough that they'll assemble together into a single sequence, but other parts won't. Smart sequencing will help with that, but it's not panacea. You can't right now sequence from a single organism, no. Not with smart sequencing. Now, theoretically, that's possible because, as you'll see here, the way smart sequencing works is that you're actually sequencing single molecules. So on the chip, you have these little tiny wells that each contains a single DNA polymerase. And that DNA polymerase will capture a single DNA molecule. And you will then, after you've started the reaction, you will then measure that polymerase incorporating a fluorescently labeled nucleotide. So basically you add 4 nucleotides, each of which is a different color, and you allow the reaction to proceed. This well is shaped so that it's called a zero-mode waveguide length, which basically means that it's the right size that the light that's emitted by this guy here can't actually come up here, and the light from these guys up here can't go down into here. So you can measure you measure the light of only the nucleotide that's actually being incorporated. So if you look here, you'll see that you measure over time, measure what the fluorescence in this well is, and there are 100,000 of these wells per chip. And you measure, and there's nothing happening, and then there's a pulse of yellow light there that corresponds to a C being incorporated, and then goes silent for a while, and then an A gets incorporated. And so you can interpret this to give you the sequence. Now, this is a very idealized version of this. The problem with smart sequencing is that it's not nearly as accurate. It's probably only about 80% accurate. So you have to have multiple reads that overlap to get around that accuracy. But you can have very long molecules here that can easily be 5 kb. Is actually 5 kb reads is the average read size. They can go up to 20 kb. And as I said, since you're reading single molecules, you could theoretically isolate enough DNA from a single cell to sequence it. But I don't think they do that yet, because you do actually have to put linkers onto these. So it's not quite there yet. There are other similar techniques that don't require linkers that might be coming on board. All right, so this is what happened when we did L-Torrentally. We made a 20 kb insert library. So we had to cleave the DNA, and you make these special libraries that had an average size of 7.5 kb and contained some 20 kb inserts. We ran 14 smart cells. We collected 2 million reads with an average read length of 3.5 kb. Filtered them to get rid of the ones that aren't really all that good, and threw out about half of them. So we ended up with almost 1 million reads with an average read length of 5,400 nucleotides. And then assemble those using an assembly program called HGAP, and it has a lot of parameters that you can fiddle around with. And essentially, at the end of the day, we ended up with 486 contigs, of which about half of them were quite large. The largest one was over a megabase of a single contiguous sequence. The median size was pretty small, because there were a lot of relatively small ones here. And then there were some other contigs. These singlet contigs were essentially very low coverage and probably represented molecules that were present or DNA sequences that were present in the genome at very low copy number, and so really were variants. And then these degenerate contigs were those that had high coverage and really were repeats, like the telomeric repeat. So essentially, most of the genome was contained in this 225 scaffolds. When we aligned those to the existing L-terentally genome, we found that most chromosomes had 2 to 10 contigs that we could align. And so here's an example, I think chromosome 29, where essentially this was that big 1.2 meg contig, and it covered almost all of chromosome 29, right, in a single contig with no ends in it. Chromosome 12, on the other hand, was the worst, so it was made up of about 15 or 20 contigs, and there may still be some assembly problems here because chromosome 12 has a lot of repetitive genes on it, and so still working on this, right? So what we ended up with is 32 megabases that contain 206 different contigs. I also added, particularly at the ends, because of the way the libraries are made, you get underrepresentation right at the telomere. So I was able to add back some contigs from the original L-terentally Illumina sequence. So we ended up with only 52 sequence gaps. So that's less than an average of 2 on each chromosome. What we did then was that we realigned all of the reads to that assembly, and now we ended up with several chromosomes that had no gaps across them at all. They had low coverage at each end, and so now what we're doing is we're going into those regions and we're doing local assemblies to try to fix those gaps. And then we'll essentially, I think at the end of the day, be able to get most of the chromosomes into a single contig that will go from one telomere to the other telomere with no gaps. Maybe chromosome 12 will be the one that's going to be the most difficult to look at. But you can see that all of the other ones were down in very small. We've got either 0 or 1 or 2 sequence gaps. So hopefully within another, and this sort of last step is very manual intensive. So hopefully within the next couple of months, we'll have this really pretty much finished. Okay, the other thing that you can use smart sequencing for is to detect base modifications. So as I was showing you before, you can see that you get a pattern of fluorescence. You can also measure the distance, the time that it takes between these peaks. And it turns out that if you have a modified base, the polymerase stalls a little while, and so that the gap between the bases increases over than what it would normally be. So that will tell you that there's a modified base. So rather than this being a T, it would be a modification of a T. In trypanosomes, we've used this to find a signature of base J, which I'll talk about a little bit more after the break. And so we can now map the position of all of the J's within the genome. And so if you zoom in, this is, I think, chromosome 25, I think, and I've zoomed into a region of it here, and we can see that there's a J here, and a J here, and a J here, and a J here, there. So we were actually able to measure, to map the exact position of all of the J's. We haven't found any other modifications so far. There's a hint that maybe there's some methyl Cs, but I'm not entirely convinced of that. So we haven't finished going through the data on that yet. And the final slide before the break is actually I've got a couple more slides after this, sorry, is single-cell sequencing. And I won't say anything other than there are techniques now available, at least for mammalian cells, for isolating single cells and making DNA and RNA libraries out of those single cells. And that there are some efforts now being started to do this with Leishmania. I think that Jean-Claude Dujardin is probably the furthest along here. I just talked to him earlier this week, and they haven't quite got to that stage yet. They can isolate single cells, but they can't get the amplification step to work accurately enough that they don't end up with a very patchy coverage. So that's really going to be the problem, I think, for Leishmania, is that the GC richness means that you've got non-random amplification. Okay, so the last slide that will be helpful this afternoon is where do we find the data for this? Well, most of you have probably already gone to TriTrypDB, which is where you should always go first to find genome data for the trypanosomatids, including Leishmania. You can go to GeneDB, which is maintained at the Sanger, which is where the data will actually go before it gets into TriTrypDB, at least the sequence data, because what happens is that the sequence data goes to GeneDB and is curated there, and then periodically that gets updated to TriTrypDB. And you'll notice in GeneDB that there will be cases where it'll say that there has been changes in this gene that are in GeneDB that aren't in TriTrypDB at that time. There's a South American database called ProtozoaDB. I'm not certain whether that's still even live. You can go to GenBank, of course, because all of this sequence will be at GenBank, but it's not set up, in my opinion, it's not set up in a way that's as easy to use as TriTrypDB. It's got a lot of data there, but it's got a lot of noise as well. And it's got multiple copies of a lot of data, so it can be difficult to sort out. You will have a chance to fiddle around with GenBank this afternoon, though. And then there's another database called TDR Targets, which we're going to have an exercise on this afternoon, which really doesn't have, it amalgamates the data, but it's organized in such a way that you can make a lot of powerful queries, and it's really designed to help people select targets that they want to make drugs against. So we'll talk about that this afternoon because you're going to do an exercise on this. This is the current data sets in TriTrypDB, and so you can see that there are a large number of genomes, and then there's information from RNA-seq and microarrays and proteomics and a little bit of ChIP or ChIP-seq and some SNP data and some different high-throughput sequencing isolates. You'll notice that many of these genomes don't currently have gene annotations on them. We're working with Steve Beverly and the WashU sequencing group and the Sanger Center to annotate all of these, and so probably by the end of the year, most of these genomes will have annotations associated with them, which I will warn you will not be 100% accurate and will subsequent to change. Whoops. And so that's where I want to stop right now. Come back after the break and we'll talk about RNA-seq.